Hello everyone! Today's video will be focused on some techniques used by scientists which involve the use of DNA. These include the use of recombinant DNA and a technique called PCR. Nowadays, genetics is one of the most important branches of biology. It can provide so much important information, and it is a branch that has evolved exponentially through time. One way to use genetics in our favor is by using genetic engineering techniques. Genetic engineering may sound odd. You hear about engineering and you think about buildings or vehicles and so on. However, engineering can be performed on the genetic level due to our incredible advances in science and medicine. So what is the purpose of genetic engineering? Basically, it is to manipulate genes using technology. Recombinant DNA is a mixture of genetic materials from different sources. This way, you can use the genetic material from different DNA strands or even from DNA belonging to different species, and then you combine them. This recombinant DNA is then placed in a host, so when that new strand of DNA is transcribed and translated, it creates a new protein which the host would not normally produce. Imagine having a car that is quite old and doesn't even have an audio system, so you can't hear any music or radio. If you go and buy a second-hand audio system that was being used in another car, and you fit that into your own car, you now have a fully functional audio system where you can listen to music, but it wasn't originally there. So you are combining pieces from two different sources to achieve your goal, which is to listen to music while you drive. That is the theory with recombinant DNA. So for this process you need several ingredients, so to say. First of all, you need a plasmid vector. In a previous video, we mentioned that bacteria have not only their own chromosomal DNA, but they also have plasmids, which are circular pieces of DNA which replicate on their own without input or interference from the chromosomal DNA. This is an important characteristic as it is what allows plasmids to be used in genetic engineering. They are called vectors as they are only used to carry the desired DNA into the host, so the original genetic material of the plasmid is irrelevant. The other ingredients for this process are the desired DNA segment, which depends on the protein that the scientists wish to synthesize, and the host cell where the protein synthesis process can occur. The desired DNA segment is inserted into the plasmid's DNA strand, and then the plasmid can be placed inside the host cell, which will produce the protein that we're after. The DNA in these plasmids usually helps the bacteria to become resistant to certain antibiotics. An important use of this technique is the synthesis of insulin. Insulin is a protein that is needed to control blood sugar levels. People who suffer from type 1 diabetes are unable to produce this protein themselves, so they regularly need to inject insulin. So the process of producing insulin through genetic engineering is the following. First, a plasmid is extracted from bacteria and a small section of it is cut out using special enzymes. Second, the DNA sequence which codes for the human insulin gene is then inserted in the plasmid. Third, the resulting plasmid is then inserted in a host, such as a new bacteria. Fourth, the bacteria will then rapidly divide, which means that protein synthesis will occur, and therefore, a lot of insulin will be produced in the process. And fifth, that insulin can then be purified and sold, ready to be used. This process is of course done at a very large scale, so it's not one plasmid and one bacteria at a time, it's loads of them at once. So how do we choose the bacteria that will become the host for this process? Basically, you start by inserting a DNA segment in the plasmids and placing those plasmids with a few different bacteria in the same environment. This process is called ligation, as it uses enzymes called ligases. Then you give that mixture a heat shock, which causes the bacterial membranes to become more permeable to DNA segments and plasmids. This way, the bacteria will take up the plasmids. Then, you place them all on an antibiotic plate. As plasmids help with antibiotic resistance, the bacteria that have taken up the plasmids will be resistant to the antibiotics on the plate, and the ones that didn't take up the plasmids will die. The resistant bacteria then start dividing and forming a colony of genetically identical bacteria, all containing a plasmid with a desired DNA segment. This process makes sure that the right plasmids and the right bacteria are used in order to obtain the desired protein without any issues. Another important technique used in genetic engineering is called PCR, which stands for Polymerase Chain Reaction. So as you can deduce from the name, it will use an enzyme which should be very familiar by now, DNA polymerase, and will cause a chain reaction of some kind. This technique is used to make several copies of a specific section of DNA without the need for vectors or plasmids. This technique uses only one source of DNA. So let's introduce the ingredients for this process. The DNA template, which contains the target DNA segment, DNA polymerase, which is responsible for forming new DNA strands from a template strand. Taq polymerase is the name of the most common DNA polymerase enzyme. As you know, enzymes are sensitive to heat, but some are more than others. Taq polymerase is an enzyme which belongs to the DNA polymerase group of enzymes, and it is especially resistant to heat, which is important as heat is used for this process. 2 primers. These are very short single-stranded sequences of DNA, which are complementary to the beginning of the desired DNA segment. So if you imagine that the desired DNA strand starts with AATCG, then your primer would be TTAGC. You need 2 primers, as DNA is a double helix, composed of 2 complementary strands. Without the primers, DNA replication would not occur. Then, you need the building blocks, or the nucleotides, which will be added to each new strand of DNA being produced. So your adenines, thymines, etc. And finally, you need a buffer. So basically, a chemical solution which provides the perfect environment for the PCR process to take place in. This process is performed on special PCR machines, and it is usually made up of between 20 to 40 cycles. Don't forget that each time you replicate one strand of DNA, you get 2. So on the first cycle, you have 2 resulting strands, and then on the second you get 4 strands, and then 16, and so on. So it is an exponential increase. So you might think that 20 cycles isn't much, but when you think about it, 20 cycles means you'll have over 1 million copies, which is a lot. So during this process, the first part of each cycle is to raise the temperature enough to denature the DNA strands, so the 2 complementary strands are separated by the heat. Then, the primers bind to each template's DNA strand, and then the Taq polymerase binds the remaining nucleotides to form 2 new strands of DNA. This is one cycle, so this is all repeated between 20 to 40 times, depending on what the DNA is going to be used for. The whole process is controlled by the PCR machine, called a thermocycler, so you can just set it up and leave it to do its thing for a few hours, and when it's finished you have over 1 million copies of that target DNA segment. Let's just talk about each stage of the PCR process. The first is called denaturation. This is when the temperature raises to around 95 to 98 degrees Celsius, causing the DNA to denature. Then, there is annealing. The temperature lowers to around 55 degrees, so that the primers anneal to each template DNA strand. The perfect temperature for this step depends on what segment is being amplified and what primers are being used. The final step is called extension. The temperature of this step depends on what polymerase is being used, but if Taq polymerase is the one, then the temperature is raised to around 72 degrees. In this stage, the nucleotides bind to the new strands of DNA. To find out how many copies you get during PCR, you just do 2 to the power of n, where n is the number of cycles of PCR. So if you've got 20 cycles, you do 2 to the power of 20. That is the end of today's video. Check out our Patreon for exclusive content that will take your studying to the next level. I hope to see you again next week. Bye bye!